Welcome to Computational Genome Biology. Today, we're diving into a disease that's often called the silent killer, ovarian cancer. For decades, it was a total medical mystery. But by decoding its genetic blueprint, scientists have found its hidden weaknesses, and that knowledge is completely changing how we fight it. We're going to follow the clues, from its very surprising origin all the way to the revolutionary treatments this has unlocked. So to really grasp why this is so important, you just have to look at one number: over 206,000. That's how many women died from ovarian cancer in a single year. This isn't just some abstract statistic. It's a massive global challenge, and it's really the driving force behind the incredible science we're about to unpack. All right, here's our roadmap for today. First, we'll look at why this disease is such a sneaky opponent. Then we'll put on our detective hats and trace it back to a source you might not expect. After that, we dive into the genetics, see how that leads to amazing new treatments, and finally, we'll look at what's next on the horizon. Okay, first up, let's talk about the core challenge. We call it ovarian cancer, but honestly, that name itself is a little bit misleading and it hides a much more complex story. So here's the first huge hurdle. When people say ovarian cancer, they usually mean epithelial ovarian cancer, or EOC. And the absolute key thing to understand is that it's not one disease. It's really a group of different diseases, and each one has its own unique molecular fingerprint. What makes it so tough is that there's no good screening test for it. The early symptoms, things like bloating or some abdominal pain, are so vague and common that they're really easy to just brush off. And the tragic result of all this? Over 3/4 of women are diagnosed at a very advanced stage, when the cancer has already spread. And this visual, it shows you exactly why it spreads so effectively. See, with other cancers, like in the breast, the cancer cells have to work hard to break through barriers to escape. But the ovary and fallopian tube, they're basically inside out. This means that right from the get-go, cancerous cells can just flake off and seed themselves all over the abdominal cavity. It's a huge head start for the cancer. Okay, so this cancer spreads super easily, right? Well, that created this massive mystery for scientists. Where was it actually starting from? For the longest time, everyone just assumed, it seems obvious, right? Ovarian cancer starts in the ovary. But the evidence just wasn't adding up. Researchers were looking for the earliest signs, the precursor lesions, in the ovary itself, and they were coming up empty. It was a huge, frustrating puzzle. And then, after years of really painstaking detective work, the answer turned out to be hiding in plain sight. The real origin point for the most common and aggressive type of ovarian cancer wasn't the ovary at all. It was right here, in these delicate finger-like ends of the fallopian tube. So let's walk through how this actually happens. It all begins with perfectly normal cells in the fallopian tube. Then, something kicks off the process, maybe some inflammation, and the cells get a critical mutation in a gene called TP53. Think of it as disabling the cell's main security system. This creates a pre-malignant lesion, which then grows into a full-blown invasive cancer. And because of that inside-out anatomy we just talked about, those new cancer cells can immediately fall onto the ovary, making it look for all the world like the disease started there. Okay, that discovery was a total game changer. It finally told us where to look. So now we're going to zoom way in, right down to the genetic code that's driving this whole process from the inside out. I really want to pause on this for a second because it is so important. A mutation in the TP53 gene isn't just a common feature of high-grade serous ovarian cancer. It's found in over 96% of them. It's the molecular driver. It's the absolute first domino to fall. TP53 is often called the guardian of the genome. And once that guardian is taken out, all hell breaks loose inside the cell. Now, to get the next part of this story, we have to talk about a process in our cells called homologous recombination, or HR. Think of it like this. HR is the cell's elite, high-tech repair crew. They're the ones you call in for the absolute worst-case scenario, when both strands of your DNA ladder get snapped in half. HR is incredibly precise, and it's what keeps our genome from falling apart. Okay, now here's where we find the cancer's weak spot. It turns out, in about half of all high-grade serous ovarian cancers, that elite DNA repair crew, it's broken. The system is just offline. We call these tumors HR deficient, or HRD. The other half have a working system, and we call them HR proficient. And believe it or not, this one single difference, whether that repair crew is on the job or not, turns out to mean everything when it's time to treat the cancer. So what's actually breaking this repair crew? Well, It's often mutations in specific genes. You've probably heard of the most famous ones, BRCA1 and BRCA2. As you can see here, a woman can inherit a faulty copy of one of these genes that accounts for up to 20% of cases. Or the mutation can just pop up on its own inside the tumor. And it's not just BRCA. There's a whole network of other HR genes that can also be culprits.
Let's really hammer this point home, because it's the whole ball game. HRP tumors, the ones with working repair crews, are tough. They can fix themselves up after we hit them with treatments. But HRD tumors, they have a fatal flaw. Yeah, their broken repair system makes them genetically chaotic and super aggressive, but it's also their Achilles heel. It's a weakness that we can now cleverly exploit.
So the billion-dollar question becomes, how do we actually attack this weakness? Well, this deep genetic knowledge has led to one of the single biggest breakthroughs in modern cancer therapy, and it's a truly elegant solution. The question for scientists became brilliantly simple. If the cancer cell already has one broken DNA repair system, what happens if we take out its backup? And the answer is a class of drugs called PARP inhibitors. These drugs block a different, less important DNA repair pathway. Now, in a normal cell, that's no big deal. But in an HRD cancer cell that's already limping along without its main repair system, blocking the backup is catastrophic. It's like the cancer cell is trying to walk with a broken leg, and PARP inhibitors come along and kick its crutch out from under it. The cell's DNA just shatters and it dies. This amazing concept has a name: synthetic lethality. And the results? I mean, they're just staggering. Look at this data from the famous SOLO-1 trial. For patients who had this specific genetic flaw, taking a PARP inhibitor kept the cancer from coming back for a median of 56 months. The group taking a placebo? Less than 14 months. That's not a small improvement. That is a nearly 4-year difference. It's a total paradigm shift. So today, PARP inhibitors are a cornerstone of treatment for many of these patients. But the revolution doesn't stop there. We're seeing other amazing smart drugs, like antibody-drug conjugates. You can think of them as guided missiles that deliver chemo right to the cancer's doorstep. For rarer types, there are other targeted drugs like MEK inhibitors. The main takeaway here is that molecular testing of the tumor is no longer just a nice-to-have, it is absolutely essential for picking the right weapon for the fight. The progress has been absolutely incredible, but the job is not done. So let's finish up by looking at what the future holds and the really big questions that researchers are trying to solve right now. So here's the battle plan for the next decade. First, we need to standardize all the genetic tests so that every patient gets the most accurate diagnosis. Next, the goal is to develop real-time trackers, maybe from a blood test, to see when a treatment is starting to fail, way earlier than we can now. Then we need to crack the code of the tumor's neighborhood to finally get immunotherapy to work. And the ultimate holy grail? Creating a simple blood test that can find not just this cancer, but many cancers, early enough to be cured.
And that really brings us to this final, powerful question. The story of ovarian cancer research is a story of turning deep science into therapies that give people more time. The progress has been just breathtaking. So as we get even better at reading and targeting the genetic blueprint of this disease, you have to ask, are we watching this silent killer slowly being turned from a deadly diagnosis into a manageable chronic condition? It's a question filled with hope, and it's being driven by incredible science. Thanks for watching. Please like and subscribe Computational Genome Biology.